The sickle cell trait is also apropos here. The Hardy-Weinberg formula gave an most frequently, selection will work against mutation, as the majority of the mutations are, but if a particular mutation, 8.3, meiotic drive and migration pressure. Besides directional selection, natural and artificial, and mutation pressure, meiotic drive and migration pressure are also responsible for shifting the gene frequencies in a population out of Hardy-Weinberg equilibrium. 1. Meiotic drive. The Hardy-Weinberg concept of an equilibrium population assumes that alleles will segregate at 1:1 ratio at meiosis and all gametes in the pool have equal chances of fertilizing one another. Hence, if gametes of genotype A1 have more success rate in fertilization than gametes of genotype A2, the frequency of A1 in the population would increase as frequency of genotype A2 decreases. Such a mode of bringing about potential evolutionary change in many respects is designation as meiotic drive. Normal segregation ratios can be influenced by a number of factors. The most widely studied example is the segregation distorter SD locus in D. melanogaster. A plus SD heterozygous male transmits to his offspring many SD alleles than plus alleles. A high frequency of SD allele might be expected in natural populations, however, the allele remains uncommon and its frequency was found less than 13.0.1. Thus, some additional selective pressures might appear to counteract meiotic drive at this particular locus, presumably by agents that act against SD-carrying flies. The existence of meiotic drive is not easy to demonstrate experimentally. However, its specific role in evolution is still unassessed. 2. Migration pressure. The most evident way through which the gene frequencies can be changed in a population is the introduction of new individuals with new genotypes in the population. This process is known as migration pressure. The effect of migration pressure can be expressed mathematically as follows. In a population X, a particular gene is present with a frequency Qx. A certain number of individuals from the population now migrate and join the population Y in which the gene in question is present at frequency Qy. The immigrant, thus, comes to represent some fraction m of the total number of individuals present in the expanded version of population Y, and a new gene frequency, Qe, becomes established. The value of Qe will be equal to the contribution made by the immigrants Qxm plus the contribution made by the original population Qy1m, where 1 meter represents the proportion of non-migrants. 9. Genetic drift Sewall-Wright effect. The American scientist Sewall-Wright 1930 explained the fluctuation in gene frequency in a small population in his essay entitled Effective Population Size. According to genetic drift, gene frequencies in a small, non-randomly mating populations fluctuate purely by chance. If population is small, then there will be fluctuation in gene frequency. Genetic drift can be explained by tossing a coin. If a coin is tossed for large number of times, then chances of getting heads and tail would be equal and the standard error is low. However, if the coin is tossed only few times, then standard error will be high and chances of getting head or tail any number of times. The drift is viewed as a stochastic process, i.e. a series of steps, and at each step the movement may The drift is viewed as a stochastic process, i.e. a series of steps, and at each step the movement made at random is directional or non-directional. 14. For example, suppose a small population of black hamsters having only one pair of individuals. The first individual has M and the other genotype M. If they produce only 2 offspring, then the chance of genotype M of first offspring will be 0.5 and the second offspring will also be 0.5. The chance of having genotype M for both offspring will be 0.5 times 0.5 equals 0.25. In this condition, the mutant allele, M, will be lost forever. Similarly, if the chances of both offspring will be M, then the allele M will get the chance to express its character in the next generation. So, genetic drift may shift the population towards losing or fixing a mutation only by chance. Gene frequency will continue to fluctuate in a small population until one allele is lost and the other will be fixed. Salient features of genetic drift. 1. It is an evolutionary force operating in small populations. 2. In a small population, heterozygosity tends to change to homozygosity. 3. Genetic drift tends to preserve or eliminate genes irrespective of its adaptive value. 4. Genetic drift may fix some non-adaptive traits in small population. 5. Genetic drift results from variable sampling of the gene pool in each generation. 6. Gene frequency in small population changes by chance because of sampling error or bottleneck effect. 7. Small population that is isolated from the large population possess some unusual characters not shown by parent population. The effects of genetic drift observed in small population are as follow. 1. Variation among populations are deemed by fixing or eliminating certain alleles. 2. Fixation or elimination of gene mutation. 3. Fixation of unfavorable, neutral or favorable characteristics in populations. 4. Increase variations between small populations but not in any particular directions. Extinction of certain species, which are left with small populations, is known to be due to genetic drift, when lethal mutations are fixed, e.g. the extinction of passenger pigeon and cheetah was due to the fixing of lethal mutation by genetic drift. Generally, population of carnivores are small and can be easily influenced by genetic drift. In some human tribes, 15, which have the culture to marry within their own communities, are also vulnerable to the effect of genetic drift and may accumulate lethal mutations. 9.1. Bottleneck effect. A population may be formed of thousands of individuals, but in the next season only few individuals may survive. The few individual forms the progenitors for the future generation of the population. Figure 1. So, bottleneck effect is a phenomenon found in animals that follow seasonal cycle of their active life and remains in active dormancy for some time, like amphibians, insects and various invertebrate species. Figure point 1. Bottleneck effect. During breeding season their populations are very large, but as the adverse climate arrives most of their populations die and only few individuals manage to survive by undergoing diapause during adverse environmental condition. This small surviving population grow and reproduces in the next favorable season by way of genetic drift. The period of crisis with low population density in the species cycle represents the bottleneck. This may result in the elimination of certain alleles. The loss of allele may differ in different demes depending on chance. 16. Accidental changes in the genetic makeup of such bottleneck populations will therefore determine changes in the larger population to be derived from them, and these changes will generally be non-adaptive in character. For example, the Arctic hare periodically reaches a peak of population at which disease decimates the population. There follows a no rabbit year, and then several years of recovery. Accidental changes in the gene frequencies during the year of scarcity must have an intense effect upon the populations of years of abundance. The same thing must be true for the lemming, a small rodent of the Scandinavian mountains. Every few years its population reaches a prodigious level, and epidemics do great damage. The lemmings then migrate to the coast, plunge into the sea, and swim until they die. The population is re-established by few which remain in the mountains. Similarly, those many species which have small overwintering populations must show bottleneck effect. 9.2. Founder effect. When a few individuals or a small group migrate from a main population, they carry only a small amount of parental gene along with them. In the small migrant group, some genes may be absent or occur in such low frequency that they may be easily lost. The unique frequency of genes that arise in population derived from small bands of colonizers or founders has been called the founder effect or founder principle. Figure 2. Figure point 2. Founder effect. 17. The principle was proposed in 1956 by Harvard evolutionist Ernst Mayr. The founder principle emphasizes the conditions that support the process of Sewell-Wright's genetic drift. For example, in most part of the North American Indian tribes, the population lack IB gene that decide the B blood group. However, in Asia, which is the ancestral home of these populations, have widespread IB gene in the population. The ancestral population of Mongoloids which migrated across the Bering Strait to North America might possess very small gene pool. Accordingly, it might be possible that the population of prehistoric immigrants did not carry blood group B. It may also be possible that only few individuals of the migrant carried the IB gene but they failed to leave descendants of blood group B. This can be explained in terms of genetic drift. Most of the North American Indians possess only blood group O, or contain only the blood allele I. With few exceptions, the North American Indian tribes lost not only blood group allele IB but also the allele that controls A blood group IA. The loss of both alleles, IA and IB, by chance defies credibility. It is now believed that some strong selective force led to the rapid elimination of the IA and IB genes in the American Indian populations. If this is true, it would provide an impressive example of the action of natural selection in modifying the frequencies of genes in a given population. 10. Summary. A population refers to a group of individuals of one kind or closely related species with no barriers to exchange of genetic material in a given area at a given time. There are two types of populations, namely unitary and modular populations. In unitary populations, each individual is derived from zygote and the growth of such individuals is determinate and predictable. Modular population includes those organisms which develops from a single zygote and serves as a unit module, forming a branching pattern. The sum total of genes of all the individuals of a Mendelian population constitutes gene pool. The gene frequency refers to the proportion of an allele in the gene pool as compared with other alleles at the same locus with no regards to their distribution in organisms. 18. Hardy-Weinberg's law states that the relative frequencies of various kinds of genes in a large and randomly mating population tend to remain constant from generation to generation in the absence of mutation, natural selection and gene flow. The formula P plus Q2 equals P2 plus 2PQ plus Q2 expresses the genotypic expectations of progeny in terms of gametic or allelic frequencies of the parental gene pool. The shifts or changes in gene frequencies can be produced by a reduction in population size, selection, mutation, genetic drift, meiotic drive, and migration. Natural selection is generally believed to determine the relative frequency of alleles in a population. A principal pattern followed by selection in natural populations is that of directional selection. Another type of selection is artificial selection, which is simply a selection program devised by humans rather than managed by natural situations. Mutation is an evolutionary agent, and mutability is a required property of the genetic material if evolution is to occur. Besides directional selection, natural and artificial, and mutation pressure, meiotic drive and migration pressure are two agents that can shift gene frequencies in a population out of Hardy-Weinberg equilibrium. In small, non-randomly mating populations, gene frequencies are found to fluctuate purely by chance. Smaller the population, larger will be the fluctuation in gene frequency known as genetic drift. Bottleneck effect is a phenomenon found in animals that follow seasonal cycle of dormancy and activity, such as insects, amphibians, and a large number of invertebrates. In founder effect, few individuals migrate from the main population, only a limited portion of the parental gene pool is carried away. 19. 1. Paper number